Eukaryotic cells contain an internal membrane system known as the endoplasmic reticulum, or ER. The endoplasmic reticulum is where lipid components of the cell membrane are assembled, along with many proteins and other products. Many of these products are exported from the cell to other organelles.
There are two types of endoplasmic reticulum, rough ER and smooth ER. Both types are present in plant and animal cells. The two types of ER are not joined together but separated. Cells specializing in the production of proteins will tend to have a larger amount of rough ER, while cells producing lipids and steroid hormones will have a greater amount of smooth ER.
The smooth endoplasmic reticulum, or smooth ER, is an organelle found in both animal and plant cells. The main function of the smooth ER is to make products like hormones and lipids. It also distributes those products throughout the cell and to other places in the organism. Smooth ER also regulates calcium ions and processes toxins. It is described as smooth ER to distinguish from the rough ER, which has ribosomes for protein synthesis on its surface. Smooth ER consists of a long network of folded tube-like structure. You can think of it like a pipeline with a lot of twists and turns. The interior of smooth ER is called lumen, which is enclosed by a membrane, just like the membrane that encloses the entire cell. The number of smooth ER units in a cell depend on the type of cell and what its manufacturing needs are. These units are situated in the cytoplasm and are sometimes connected to a unit of rough endoplasmic reticulum. Typically, smooth ER is placed away from the nucleus. Smooth ER is a transition area where chemicals that the cell has manufactured are stored in the lumen for transportation elsewhere in the cell. Pieces of smooth ER, called vesicles, pinch off from the smooth ER and travel other places in the cell to transfer their content. You can think of a smooth ER as a factory producing many of the products that a cell needs to function. Exactly what it makes depends on the type of cell. Imagine two factories in two different locations. One is in the middle of a farm country where corn grows as far as the eye can see. The other is on the coast of the ocean where fishing is the main industry. The factory in farmland manufactures corn syrup to be used in a number of packaged foods. The oceanside factory produces canned fish products. Each factory makes and distributes the type of product that makes sense for its location. Smooth ER also makes or distributes products according to its location. In muscle cells, it releases calcium ions because they are required for the muscle contraction. In cells of the endocrine system, which is responsible for regulating hormones, smooth ER makes steroids. Smooth endoplasmic reticulum functions in the synthesis of lipid-containing macromolecules. Lipids are universal biological molecules. Large amounts of smooth ER are found in the cells that specialize in lipid metabolism. This metabolism also involves in the degradation of fatty acids, which are fundamental biological molecules and the building blocks of more structurally complex lipids. In order to be metabolized by the cells, lipids are hydrolyzed to yield free fatty acids that then are converted to acetyl. We are constantly being exposed to materials that are poisonous to us. Such materials are described as toxic. Most are naturally occurring. They might be produced by plants or animals as a means of discouraging other organisms from destroying or eating them. Some of the plants and animals are selective and others harm everything and everyone around them. That is why we have developed a way in which we can protect ourselves from the poisons. The smooth ER contains a large number of different enzymes. Among these is a group of enzymes that absorb of particular light frequencies and are known as cytochrome P450 enzymes. There are a number of cytochromes P450. All of them are different enzymes, but they almost do the same thing. They both add oxygen to compounds that they modify. There are a number of different oxidation mechanisms, and each cytochrome appears to act on a different set of compounds. There are many things that are not known about them, but we know the most important one. They are changing the chemical nature, and that's how they remove toxicity. Most of the times the toxicity of a compound is destroyed or diminished. However, in some cases, cytochrome action may actually produce or increase it. Indeed, some drugs are ineffective until they are activated by the P450 system. Smooth ER is a part of the endoplasmic reticulum. The smooth ER is a network without ribosomes. It is concerned with lipid metabolism, which is important for people's health, and it also exerts toxic substances from our bodies.
The RAF endoplasmic reticulum, or RAF-ER, is an organelle that is involved with the production, folding, quality control of many proteins and their transportation to other parts of the cell. It is an extensive membrane network of cisternae which is continuous with the nuclear membrane. It is called RAF-ER because it is studded on its outer surface with ribosomes. These are called membrane-bound ribosomes and are firmly attached to the outer cytosolic site of the ER. Part of the RAF-ER is continuous with the nucleus envelope. The Golgi apparatus is also closely associated with the ER. Still, RAF-ER is found throughout the whole cell, but the biggest concentration is near the nucleus and the Golgi apparatus. Certain cells of the pancreas and digestive tract have bigger amount of RAF-ER and produce a high volume of protein which is used as enzymes. Ribosomes on the RAF endoplasmic reticulum are responsible for the assembly of many proteins. This process is called translation. During protein synthesis, the ribosomes are the site where messenger ribonucleic acid, mRNA, which is a RNA molecule that conveys genetic information from DNA to the ribosome, builds proteins according to the specific code it carries. As the protein is produced, it leaves these ribosomes and is inserted into the RAF-ER, where it can be chemically modified. Once the protein is complete, a bit of the RAF-ER membrane breaks off to form a vesicle around the protein. It may be transported in the vesicle to the Golgi apparatus or transferred to other organelles. The RAF-ER, working with membrane-bound ribosomes, take polypeptides and amino acids from the cytosol and continuous protein assembly, including at an early stage recognizing a destination label attached to each of them, so we know whether the proteins are produced for the plasma membrane, Golgi apparatus, secretory vesicles, plant vacuoles, lysosomes, endosomes, or the endoplasmic reticulum itself. Some of the proteins are delivered into the lumen, which is the space inside the ER, while others are processed within the ER membrane. In the lumen, some proteins have sugar groups added to them to form glycoproteins. Some have metal groups added to them. It is in the RAF-ER, for example, that hemoglobin is formed. The ER lumen maintains a chemical environment that ensures that proteins are folded into the correct conformation. Misfolded proteins are useless and may cause problems if they are detected as foreign structures by the immune system of the body. Newly synthesized proteins are quickly associated with ER chaperone proteins and folding enzymes that assist in the folding of the proteins into their correct conformations. It is also in the lumen that an amazing process of quality control check is carried out. Proteins are subjected to a quality control check, and any that are found to be incorrectly formed or incorrectly folded are rejected. These rejects are stored in the lumen or sent for recycling for eventual breakdown to amino acids. A type of emphysema, a lung problem, is caused by the ER quality control section continually rejecting an incorrectly folded protein. The protein is wrongly folded as a result of receiving an altered genetic message. A form of cystic fibrosis is caused by a missing single amino acid, phenylalanine, in a particular position in the protein construction. The protein might work well without the amino acid, but the very exacting service provided by the quality control section spots the error and rejects the protein retaining it in the lumen of the RAF-ER. In this case, the customer, the person with cystic fibrosis, loses out completely due to high standards, when a slightly poorer production would have been better than no production at all. The RAF endoplasmic reticulum is a membrane network studded with ribosomes. It is involved in the production, folding, quality control, and the transportation of proteins.